All right, so what ChatGPT doesn't know about sterilization? ChatGPT doesn't know much about sterilization. It seems like there is not much training material, so it failed big time with many questions. And what I'm going to present today is about the pressure we have on EO sterilization. So this is where everything is starting. Maybe 5 years ago, this became more and more. And then, as a response to this pressure, some controversial validation approach I want to present to you, which is used, and of course, then also alternatives to ethylene oxide. So first of all, acknowledging the risks with EO. So there is definitely risks for staff and patient. Okay. It's cancerogenic. It's an IARC group 1 cancerogen, which means that there is no doubt about it, that it's cancerogenic by exposure, by inhalation, and with direct body contact. So it means that in the facility and surrounding areas, there is potential risk if it gets into contact. And the direct body contact is then for the patient. There is rest of EO on the medical device, and it's also risk that you must address, okay? Then, of course, it's also toxic and mutagenic. So risks, again, handling it in the facility, but also for the patient. There is some toxic risks with EO, so with ECH. And it's also explosive, so there must also be addressed. It's also definitely an important risk. So now the question is, are these risks manageable long term? Okay, so I believe yes. I believe they are appropriately and can be appropriately addressed. It is definitely cancerogenic, but that's not a reason to 100% ban it. Okay, so just one example, similar to vinyl chloride used for PVC production. It's still there. It will be still there, but it's managed. Okay. And we managed it by explosive, by several technological mitigations. We address this by several limits and so on we must keep. So from my view, this is manageable, okay? So now the public and regulatory attention. So in the US, there were big site closures. I believe it was in 2000, 2021, a big site closed. And this had huge issues with medical device manufacturers. So quarterly financial warnings from very big companies and so on. So they really this really caused a lot of trouble. The site closures was because of surrounding areas, discussions about high cancer rates, and so on. I'm not expert for the US situation, okay? But this is what happened, and this is what caused a lot of attention. Then there are quite a few litigations also in the US, so companies that paid several or $100 million of dollars. And there is ongoing EPA rulemaking. It means that EPA made some stricter rules in 2024. And now these rules are still discussed, okay, and are made here in the attachment. You will find a source where Congressman it's just a comment 2 or 3 weeks ago, Congressman commented on this, on the EPA that wanted to make the rules a little bit less challenging. They insisted to keep them on the level from 2024. So it's still discussed, and there are still, let's say, risks, not clear where it's going to go. This the rules. So in Europe, we have no comparable closure trend, and we have established EU and national controls. So I'm asking this a lot, okay? I talk with many people from Europe mainly, also from the US, but mainly in Europe. And there is not much pressure from the authorities to, for example, reduce EO emissions or increase worker safety and so on. But definitely, the topics EO residuals, worker safety, emissions, and permitting, it remains sensitive. And I can give you a few examples also in Europe. For example, currently two of my clients from Europe, they're in the same country and they are both extending their EO sterilization capacities. They built a new chamber. Um, Even though it's in the same country and pretty much the same case, it's different authorities who are checking them. So for one, it's relatively smooth. I mean, they still do have to keep the rules and so on, but it's relatively smooth. And the other one has to do like tons of paper and has already a year of delay because of the permits. So also permitting is it's getting more difficult because EO, cancerogenic and so on, it still is could be a red flag. Okay. So there are also risks definitely in Europe. Or I'll give you another example. I worked from the manufacturer who was not allowed to sterilize a new device with EO. It was for neonates, and this authority pretty much forced them to search for another option. Okay, so the regulatory risks remain. And then the supply chain dependency. So a company's entire product portfolio may depend on EO sterilization. As I said before, that site closes and a very big company has less sales. I also know companies that are sterilizing everything, for example, in-house through one EO sterilization site. So if they have no issues, for example, by regulatory pressure, changing the sterilization site can take months. So in this case, sterilization can become very business critical, and I know quite a few such cases. This is when you, as a consultant, often are getting into it.
Hey, Manuel, I wanted to ask you, this is very clear what you're presenting. But I wonder if there has been any discussion in terms of risk-benefit ratio from a supply chain perspective, patient access to these medical devices. Because again, if somebody said no more EO sterilization, we would create a crisis that would actually put patients at higher risk
Mhm.
than if we had a controlled approach, a
Absolutely.
risk-based approach. So, has there been any discussion from a risk-based perspective?
Yes, a lot. So this is discussed definitely a lot in the US mainly, in Europe not.
Mhm.
But there is there are several discussions about exactly this point, and many people then advocating for EO and saying we would have big risks if we would put it down.
So I just like to see a show of hands, like how many people in the audience are well familiar with sterilization versus or put in like your level of familiarity on a scale of 1 to 10. 10 being I'm a sterilization expert and one being um, Sean. You can. That's interesting. Oh, we got a 0. We got a sub Sean. Okay.
So how many sterilization experts? I don't see it. Can you tell me?
I don't think I've seen a 10. Um, I I'm gonna average it at 6 or 7.
All right, that's great. Very good. All right, so EO is indisplaceable in the medium term, and that's very clear. There are 20 billion devices produced, sterilized with EO per year alone in the US. So 20,000 million. Okay. And more than 50% of the single-use medical devices worldwide are currently still sterilized with ETO. And here's the source, Rutherland and Weber, 2023, also saying ETO is irreplaceable for years, and also discussing, Daniel, what you just said, risk benefit, risk-benefit issues, and so on. Okay. So why there is no like for like replacement? So it's a combination of things. So first of all, there's a very high penetration into packaging, lumens, and difficult to sterilize location, and very dense pellet slots. Okay, it works very well. It penetrates very well and better than other modalities. Then there is a high material compatibility for a very wide range of materials. So there are not many materials that have an issue with EO sterilization, opposed to all other or pretty much all other modalities, where you have for certain materials certain issues. And then there are many sites for contract sterilization available. It's when I work with customers and they think about alternatives with a new device. I mean, this is often an important argument saying, well, there is a contract sterilization facility, but it's on the other side of the globe. Okay, basically. So this is a big advantage for EO. And it's difficult to build that up, you can imagine. Okay. And then there are also many capacities for building your sterilization plants. So if we say now we want to sterilize this 20 billion devices very soon with other modalities, well, there must be also companies building that capacity. And this is simply not existed and also discussed in this paper here. So what I believe is that and I could see this differently in a few years, but what I believe now is that EO will likely stay for decades, especially for sensitive, difficult to sterilize, low-cost products. So, for example, sets, very complex sets that are relatively low cost with very different materials inside. This procedure packs, for example, that are sterilized in big pellets and very dense and 100 different materials or hundreds. These will be the last devices that will be not EO sterilized, and it's hard to imagine how they would be EO sterilized. It would be a big project to get all these materials changed and so on, or to find a modality that works.
May I just jump in?
Yes,
Yeah.
please.
With regards to alternative methodologies, I actually had a few years ago an opportunity to do a clinical evaluation about the alternative possibilities, like using ozone and hydrogen peroxide. And I fully recognize that EO is a low-cost method of sterilization, but at the same time, it does have toxicity. Products that are EO sterilized need to be in a warehouse for a certain amount of time so the EO can expire. And what a lot of people don't realize is that the medical device regulation EU made actually these products more dangerous because the label on the package became even bigger, which prevented a lot of the EO from expiring. Now, you say in the future that EO will be still around for decades, but I think there is a push to find alternatives.
Mhm.
Why are you not presenting ozone or the other ones, or will you be
I
presenting
am
maybe?
definitely, I'm very
Okay,
excited about these
okay,
alternatives. And I
okay.
think we should work on this. But I tell you from experience and what I see on the market and so on, that I see this trend. And for me, it's super interesting, business-wise also. Okay.
Is the
Nevertheless,
financials blocking the implementation? I mean, there's one firm, one of the big firms is Steris, right? Which I'm sure you're aware of.
mhm.
Is the financial, the cost of the alternatives blocking the alternatives being used?
I mean, the thing is that so I started a big project where I contacted several manufacturers and presenting them possibilities for the alternatives. And most of them just had other priorities. And I totally understood this because in Europe, at least, okay, I can tell you for sure, it works pretty well with the sterilization. I don't see that this is going to be changed in 10, 15, maybe 20 years, because it works well. It's cheap. We have limits that are acceptable, that are challenged a lot. And there is a very low risk for harm to the patient, to the workers, and so on. So these risks you have often in the industry. I did apprenticeship with 17 as chemical lab technician, and we cooked some new substances that were never tested on humans, some very complex substances. So there was also a risk that they were cancerogenic or toxic and so on, definitely. But we addressed this by procedures and so on. And I think it's similar with EO. So you can address this. And the companies, so back to your question, the companies just say, I don't see the reason for me to change. Where are the benefits? I have many other projects. It's interesting. We want to do it, but then on a higher level, it's often then decided, let's do this. Let's do this again. Let's discuss this again in a few years.
Yeah, Manuel, I just wanted to also comment that when you talk about compatibility between material and the sterilant, this is important because in some cases, if we had to comply with a different sterilant, we may have to also change our the material
Yes,
that we use. So, and that brings now another set of challenges to the industry in terms of
absolutely.
cost and development. So, I just wanted to raise that.
That's a very important point. Okay, yes. But we will come to that also later. I have a few slides for the alternatives, but thanks for the good discussions. So what manufacturers do and experience right now with regards to that pressure? What are the reactions? Okay. So one is lower EO concentration cycles. Let's say lower than 400 milligrams per liter is usually something you will see. This, I think, came from an EPA, an earlier model or an earlier requirement saying that you must go under 400 milligrams, if I remember correct. So this is now picked up from sterilization service providers who are in the US, but also worldwide. They reduce their cycles accordingly. And also big, mainly US companies who are saying, well, we want to have cycles lower 400 milligrams per liter. It can reduce workers' exposure and emissions, definitely. So I also saw some data on this, and this can have positive effects, but often there is no technical need to reduce it. So what I want to say is that if you have a cycle, especially in Europe, with 600 milligrams, 650, you will be usually fine and it's still used, and there is often no pressure to reduce this. Nevertheless, this is an important trend. But what happens if I reduce the EO concentration in the cycle? So if I put inside 400 milligrams per liter instead of, say, 600, 700, then my cycle needs more time. It needs more time to kill the bacteria or the microorganisms to a certain level. And but you don't want this. You have a certain capacity, and of course, you still want to have your, for example, 8-hour cycles. Okay, so this reduced efficiency is addressed by mainly addressed by a change in the validation approach. So now very high level cycle calculation approach rather than half cycle approach. I will just briefly discuss this later. But this here, use of product tests of sterility as a reason not to inoculate the product. This is the ugly one. This is unfortunately by some companies proposed, and this will put you in trouble. And there I'm going to present quite a while and explain you what is the consequences and what is the reason why this is not a good idea. Okay. Then there are the alternative modalities, and I'm also going to have a few slides on that. And the current efforts appear to be largely driven by US-based medical device manufacturers. Why? Because there is this higher pressure. However, the alternative modalities may offer significant benefits beyond the US, as well, particularly for in-house sterilization. So this is what I'm really excited about. I see some potential benefits there for companies. Then what you also see now a lot, and this is in the US, but also in Europe, you see technology upgrades, okay? Step by step in Europe and the US now much faster. So filter and abatement systems. It's quite expensive catalyzers or also filters in the desorption holes, some dry bed filters that do recirculation and reduce the EO concentration in the desorption holes. Then automated handling during desorption. So after the EO sterilization, when the pellet is packed with a lot of EO, you should take some measure, or you can expose the operators even less if you have an automated handling. And I think this is a brilliant technology that it goes directly into a tunnel and then into a SOIT in an automated warehouse, and the operator does not touch the pallet in routine production for 3 or 4 days. Then sealed cap forklifts into the desorption hole, and then extended desorption time. So it's very interesting. So instead of, after 3 days, you would often be under the limits for the patient. For example, 4 milligram ethylene oxide per device is a limit, and you would drop under this limit after 3 days after EO sterilization, and then you could release it to the market. But still, there is EO on the pallets and so on, and of course also in the product. So some companies say, well, I want to have it 7 days in your desorption, in your control desorption. And this helps a lot then downstream also in the warehouses, or 14 days, okay, extended desorption times. And then also EO monitoring, okay, just monitoring more on the fence line or at the chimney or in the desorption, how much EO do I actually have? This is also increased. So all this makes sense and gives you more safety, definitely. But of course, these are all major cost drivers, okay? EO sterilization gets more expensive. And then just quickly backup sterilization or a second source. So pros, it strengthens your supply chain, and also it can help you in negotiations if you have a second supplier. But cons is more validations than suppliers. So you have another validation in the second site that you must do and revalidate. You have another supplier that you must manage. And what is interesting, what I find interesting is that if you have high volumes, like let's say, for example, 10,000 pallets per year or 20,000 at supplier A and the same at supplier B, well, it will be difficult for supplier B to, in case there is an issue with supplier A, to just ramp up to 40,000 pallets per year. So your high volumes are usually not 100% covered, also if you have a backup sterilization. All right. So now I'm going to present you, let's say, the usual validation approach that is accepted. And this is now a lot of diagrams and so on. I hope I don't put you to sleep, but if you watch this, if you follow this, then you will understand the most important part about sterilization. So let's say you have this is a tubing set, and on top there will be always a Tyvek or a paper, usually a Tyvek here, okay? So it's a sterile barrier. But now the sterile barrier is removed. We just have this tray here and this tubing set. Now you say, okay, I want to label this sterile. I want to have this sterile. What do I have to do? So what we use is these BIs, biological indicators. They have a Bacillus atrophius. It's called the microorganism. Around 1 million of them on one strip. Okay, so you know the exact type, you know the number of microorganisms. And these you use for the validation. It's a BI-based method, not a bioburden-based method like other sterilization modalities. Okay, absolutely not. It's based on BI. So where to put these BIs? You would put them at parts where you want to show that EO or also steam and lethal conditions are reached, because this is your indicator that is showing you I killed 1 million spores. So if you put it somewhere it will be, of course, easier for EO to kill here a spore on the surface of this tray, opposed to here in this lips. So this is a very usual place where to put a BI. Then also here you want to investigate, this is a volume inside of this tubing. Here you would also like to know, well, how much lethal conditions, how much kill do I have? And then the third BI you would maybe put here, okay? This is the so-called longest gas pathway. You close here, you can do this, and then you make sure that the EO goes through this, through the whole set to this point here. So this is a high challenge. You could also remove it and place the BI here in the middle with a T or something. But this is also something that is done a lot if you can afford it to challenge the process a bit, okay? So now we come to this diagram here, and let me explain it to you quickly. So we have here on the x-axis the EO contacting time. It means just half hour of EO contact, 1 hour of EO contact, 1.5, 2 hours. This is the EO contact. And this here is the number of microorganisms or surviving microorganisms. For example, you start at T0. If you have no EO contact, you have from the BIs this 1 million here. Okay, this is your starting point. And then the EO will reduce the population of your BI. And that's why, and here we have the probability of surviving microorganisms. So after some time, you will have one microorganism and then 10 to the minus 1, 10 to the minus 2, so a probability of 1 to 100, until you go down to 10 to the minus 6, which is the probability you usually want to have for your sterilization process, your SAL, okay? So if we would have, several data points, if we would measure after half hour, after 1 hour, after, and so on, it would more like look like this, okay? The curve. It's a log linear reduction because you have it at a certain location. You have constant conditions with regards to EO and temperature and so on. And you have this Bacillus atrophios, so just one type of microorganism and not several types that would die off differently. And then this leads to this log linear reduction, more or less, okay? And now the bioburden behaves different. The bioburden is the microorganisms you have every day in your production. And here you have, first of all, it's not in a certain position. It's distributed in the whole set. And second, you have different kind of microorganisms on it. So it will die the easy to kill microorganisms, and at the easy to kill positions, it will die off very fast. And then you will have such kind of curve, okay? A tail, it's called. This is how bioburden inactivation behaves. So you could also do some tests and measure it, and we assume now for this set, this kind of bioburden inactivation. So now what you would do first in this usual validation approach is a so-called sublethal cycle, okay? So you still have some, this is like, let's say 1.5 hours, for example, or 15 minutes better. So you would do this sublethal cycle and you would measure how much survivors do I have on this BI, on this BI, on this BI. And you would see that I had the highest number of survivors on this BI in the syringe. And this is now your worst case BI inactivation. So the EO is the most difficult part for the EO to reach, or maybe with other words, to create lethal conditions is this BI. And this you want to know. This is now your worst case BI inactivation, your rate. And this, when we come to that, how you're going to use this. Now the bioburden, you would, in the sublethal cycle, you would also do test of sterility of this product, and you would see it's sterile or it's not sterile. So in this example here, it would be definitely most likely sterile. There is a certain probability of growth, but let's say this is then sterile. And what is also interesting, this rate here that is important, and I will discuss in more detail in a minute, this rate here for the bioburden will now be not much worse than parallel to this worst case BI inactivation. Why? Because in the worst case, there are some highly resistant microorganisms like the Bacillus atrophius at this position, okay? All other positions will be less resistant, so you will have more or less a parallel curve, all right? And this is important, I'm going to tell you later. So now I'm doing a half cycle with the set here, but only this inoculated with this PI here. You could also use an internal PCD. This is possible, but you have to show that it has this rate here, all right? So you do a half cycle, you show total kill of this PI here, and then you showed 6 spore log reductions, yes? Then for your full cycle, also for your routine cycle, you double the time here. So let's say this is 2 hours. You would double it, and you would have a 4 hours EO contacting time in routine. In 4 hours, you are able to reduce to 0, and then also this probability. So to extrapolate it to 10 to the minus 6. By the way, there is also a cycle calculation approach. I mentioned it before that will lead to less EO contacting time, but I'm not going into that now in detail. So if I do this here, what I just explained, then I will have a BI better than 10 to the minus 6. If I follow this curve now, and this was the actual worst case BI inactivation, I must add a bit in my half cycle to see total kill. But if I follow this curve now, after this time here, I'm at the SAL of 10 to the minus 8 or something, okay? So, but I'm not in the BI killing business. I'm in the sterilization business. I'm interested what is going on with my bioburden activation. And for my product, the product SAL, let's say, is much better. I said here BI SAL and product SAL. The product SAL is much better than 10 to the minus 6. So if you follow this line here, it's somewhere here, I don't know, 10 to the minus 12 or so, but you don't really know it, okay? Because this was just an assumption. What you know is that this is parallel to this, and you have some safety margin. So you can tell for sure you are much better than 10 to the minus 6 for your product bioburden. And this is where many say, wait a second, I don't want a cycle with so much overkill. I want a cycle that was just 10 to the minus 6. And there are possibilities. I already said the cycle calculation approach, or you say, I don't have $1 million bucks on it. I have 10 to the 3. You could even reduce it more. So there are possibilities. But always you must take care of this rate here. This is your indicator that is showing you at your difficult to sterilize locations your die off. Okay? So this is the accepted approach. It's overkill. It's the accepted approach. And if you come up now with another approach that is more efficient, that don't have so much overkill, then you must really explain this statistically. This here is easy to explain, easy to defend. And now there is this EO validation relying on test of sterility, TOS, and it's really very much with this lower concentration EO cycles combined, because you want to address this loss of efficiency. And high risk of rejections, I wrote here. So what is done? The product is not inoculated. I'm going to show you some pictures later. The product is not inoculated. In this approach, instead, a separate internal PCD is used, or the BI is placed outside of the product. And then the comparison of the internal PCD versus the product bioburden is intended to be performed using a sublethal cycle, which I showed you before, after which sterility is demonstrated from the products, while growth is demonstrated from the internal PCD. Okay? So you say, now my internal PCD is okay, it's more resistant than this product bioburden, because here I have growth, here I have no growth. And this is the, let's say, easy logic behind, but there is a big problem with this. And this I'm going to show you in a minute. So there is no deactivation rate quantified at the difficult to sterilize locations of the device, which I showed you before. So how is this now done? You have again, same device. And now you use some kind of BI, whatever it is, some kind of PCD, and it can be a very easy to sterilize, okay? Or you place your BI here instead of the syringe. This is also possible. So now we have these situations. The BI is now relatively easy to inactivate. And this was more or less the situation before. So we can have much shorter times also, or reduce the EO concentration. And this is again the bioburden inactivation. So now we run this sublethal cycle. And as you can see, this criteria I said before, growth in the internal PCD and sterile products from the test of sterility, it's easily achievable. So it's very likely that it will be achieved. But the question is, what is it going to tell me about my SAL of the product? So you would now run the half cycle with this BI here, or with this PCD here, double it. This is now your more or less, it's now a bit simplified, but this is your full cycle time or your routine cycle time. And the SAL of 10 to the minus 6 is definitely reached for this BI, okay? You showed this, and this is okay. But the bioburden, if you look at the bioburden, it's simply, the curve here is flatter, so it takes more time, and after a while, it crosses. So you have an SAL of 10 to the minus 4 here for your bioburden, okay? Theoretically. So this test doesn't tell you much, and it can be worse also. So if you have this kind of situation, the SAL will be like 10 to the minus 2. And this is the main issue with this approach I wanted to share with you. Now, this approach, so using product tests of sterility to relate to the internal PCD, remains controversial in standardization committees. So we have lots of discussions there and are asking for a rationale, which we don't get so far. And this is really important, I often see it rejected during submissions. And I totally understand notified bodies or authorities rejecting this. And these rejections, from my view, are unlikely to change with the ISO 11135 update. I was asked 2 weeks ago, I use this approach. With the update, is it going to be fixed? I heard it's going to be fixed. And no, it's not going to be fixed. So there are still, it is proposed in a certain way in this ISO update. And you can also download it, by the way. But due to ambiguities, and I want you to have a look on chapter 8.5, which is telling about this inactivation rate I told you, but also B23, which is telling you about complex devices or devices where gas is difficult to reach. And especially in the FDIS update, I want you to have a look on these chapters and then again reflect this validation approach, with the test of sterility. Well, and then there is lack of statistical sound rationale for this validation approach, and there is a paper I also attached. So we're still waiting for a rationale. It's simply not existent. You will not find it anywhere, how at the end of the day this should add up. And now I give you this audit killer phrase. What spore log reduction or sterility assurance level did this validation actually demonstrate at the most difficult to sterilize location of the device I'm holding? You're not able to tell at the most difficult to sterilize location what is your SAL or your spore log reduction, because you did not measure it with this method. So now quickly, if your recent validations relied on this approach, the potential downstream cost if challenged are, you must repeat the validation, you do and if it fails, and this is very likely, if it fails if you put the BI to the worst case location, then you must develop a new cycle, do again a validation. And then maybe you did your packaging and product testing with this initial process. So maybe you have to do this again because now you have a process with more EO concentration or longer dwell times and so on, EO dwell times. So you have definitely high submission delays and very high costs. And, this is my main concern here, that this happens. Also for the auditors, it's not a pleasant situation. Well, and then also there is the sterility, of course, a risk for the patient. Okay, good. So we have 15 minutes left, and I wanted to tell you about alternatives. Other questions so far with regards to that? Or do we want to ask them later in the Q&A?
Yeah, and Manuel, I think that was an interesting question. I think the topic included the discussion on ChatGPT, and somebody was asking, how do I make that link? Is that something you're going to bring up relatively soon, or is this something that maybe it's apropos to be brought right now? And just a question.
Well, he did mention at the beginning that ChatGPT doesn't tell you very much about sterilization. Um, I think the person may not have heard that. Yeah. Um, I think that I'm certainly seeing a level of detail here that I didn't get from my own look at ChatGPT um, around this topic. Uh, I don't know about anybody else. This,
It, uh, and I think you're absolutely right, Sean and Manuel. I mean, when you think about ChatGPT, like any other AI forum, it's highly dependent on the input it gets from,
Yes.
the technical papers that are developed, the guidance documents. And this topic that Manuel is talking about is evolving. So ChatGPT has no ability to rationalize anything that is extremely relevant on specific aspects of this topic. And this is why the human element is still the leading force behind which way these concepts are going to go.
Yes.
So as
Yes.
soon as we start feeding into ChatGPT, you're going to see more ChatGPT reaction, which is going to be based on human input, and that's still not
Yeah.
necessarily there. I guess, yeah.
and where else are you going to have a sterilization expert come on and speak to you for free and educate you on the importance and understanding and the level of detail that Manuel is giving us today? I don't know of any other forum that is presenting this free to the public.
Manuel, the last time I remember reading the standard, there was no differentiation between um, small children and adults. In fact, humans were all in one bucket. Has anything changed there? Because really,
Yes.
yeah, okay, good.
It's a bit of a different topic now, but I am going to answer it. So there is an update of the standards, is the ISO 10993-7, and it's a very good update. It's a very good standard, and it's absolutely addressing this topic.
Thank you.
But that's a
All right.
biocomp standard that you're talking
It's a
about. Of course,
it
that
is, but it's for EO residuals.
okay. So the EO standard, I mean standards for associated with sterilization do not address the topic. So it would be really the biocomp because of course that would
But
be the
they are.
that is
So all standards are referring to this biocomp standard, and
yeah.
sterilization experts use this standard, even though it's in the biocomp family, let's say.
Yeah.
10993-7 is directly tied to EO.
Yes. All right, we have 10 minutes left. So I would like to tell you about this modality. So sterilization modalities, this is from AMI 17. And so right now we have here ethylene oxide. And what is interesting, it works by alkylation, okay? So it's a very specific chemical reaction, which works very good with proteins or DNA, but does not react much with all other with most of the other materials. And this is why it works so well and so specific to kill DNA or to kill bacteria and microorganisms, but not to damage your product. Then we have, which is also an important modality, ionizing, so radiation, gamma, E-beam, X-ray. It's very interesting, and I think for some medical device manufacturer, it's the right thing to go. I saw a very interesting presentation about E-beam 2 weeks ago, but it's ionizing, and you might have some material effects, so you must test it. And then very interesting candidates are then nitrogen dioxide, hydrogen dioxide, which we heard before, and chlorine dioxide. And they are all oxidizing chemicals. So there might be some effects with your plastics, but also with your metals and so on. Okay. So now, for me in particular, interesting for in-house sterilization, but there are also some advantages if you do this with the contract sterilization manufacturers. But in-house, I'm going to show you what I think it's interesting for many suppliers, medical device manufacturers also in Europe, is so you in your facility, you build the device, you do the sterile packaging, and then you sterilize it. This is from Steris here, for example, but basically the same with these modalities. You sterilize it on a tray without cardboard. You pack it in a cardboard afterwards. Works a little bit less than EO. It does not penetrate so well through cardboard. So you pack it afterwards and then you ship it. So you are super lean, okay? So it has a lot of advantages with regards to lead time. If you have a expensive product with inventory and so on, this can be a brilliant solution. You have a low footprint, so you don't need so much space than with EO. You have almost no desorption times. I know suppliers that wait 2 months for the product, so that could be a good reason to change. You have low risk for permits for extension or new sterilization sites. I told you before with EO, this can be a problem. Here it's less of a problem. And yeah, it reduces all these risks we discussed before with EO. So, so I think using this, especially in-house, is interesting. Like with steam sterilization, you would usually do this in-house because it's steam, it's not EO, which is cancerogenic and so on. And all these substances are definitely much less dangerous than EO. So you could think about to do this in-house. There is also E-beam is another option. I did not go into just for in-house also. Good. Now, just quickly, how would you do now this? How would you start working on this alternative sterilization modalities? So first of all, what is your opportunities, the evaluation of opportunities? So I named it before, less inventory could be a good reason, less EO residuals, less EO desorption time, so 2 months waiting for the product and so on. These are all chances or opportunities, benefits. And this is the first thing you do. And if you see here, if you have basically a business case here, then you would move forward and do this feasibility study. So as you would do this in providers who are producing or manufacturing these plants. You can do some feasibility tests. You would create a process that will most likely be able to be validated, so robust enough for your product, lethal enough. And then you would, with this process, you would then do testing of your materials and so on. And then you can see if it works for which part of your product maybe, and how does it work? Is it feasible for you? You would then do this regulatory approval, and this can take some time. So we have a lot of movement here and moving forward here, but this can take time. And this will be the next step. And you don't have to buy equipment for this. You could also do this in a certified contract sterilization facility. Start with the approval, maybe with one product, get it through. And then maybe, so stay with this external sterilization provider and maybe in parallel do this transfer to an in-house sterilization. So this is where you then start with a bigger investment and you would buy such equipment, implement it in-house. But this is when you know it works, it's feasible, it pays off, and I will get regulatory approval. All right, so just quickly, main working groups. AMI TIRs are working on this. Kilmer is working on this. International Scientific Exchange is working on this, which is basically TÜV Süd, Medtronic, and Johnson & Johnson started this. You see this bunch of people. This was 2024. And when I saw this picture here, I thought, well, all these people would not come from the States and so on and from Asia to Munich to talk about this if it wouldn't be interesting and important. So I joined also in 2025. We were much more people, but we don't have such Beautiful picture. So key takeaways, please be very, very, very critical of EO validations approaches that only rely on product sterility testing, that rely on product sterility testing instead of inoculating the medical device with the BI. And then EO sterilization will likely remain essential, but alternatives should be evaluated.
Thank you for the presentation.
Welcome.
I don't know if anybody wants to chime in with any questions. We have a couple minutes left. I would encourage you to connect with Manuel on LinkedIn. And if you are in need of a sterilization expert, I'm sure he would be happy to advise you. And anybody learn anything new today? I know I did. Personally, it was a world that I just have never thought about in any detail. So I appreciate this.
Thank you.
Nice job, Manuel.
Thank you very much.
That was a lot of detailed information that you actually put in front of us that made sense. So great job.
Thank you. Yeah, if you have questions, just reach me out. I'm very happy to answer them.
Manuel, I have one question. And it's with a current client, they have a new single-use device, and it's so, to me, as an engineer, backward, their solution. I'm trying to figure out what they're thinking, and they don't explain it well. Their packaging is way oversized. So anything from storage to, I'm thinking of 10993-7 residuals. Is there any reason? They said it's to protect the product. So to me, their product packaging is perhaps 3 times larger than it needs to be, which I would think every stakeholder in the company would be saying, why are we doing this? But it's made it through all of that. Is there any reason from a sterilization perspective that you would have a larger than normal package if everything else is done well?
I mean, it's mainly costs, and you must explain well why you do this. So you need data why smaller packaging, let's say double instead of triple size, does not work for you. You need data for this. Otherwise, I don't believe that there is a reason sterilization-wise to do this.
Okay. Yeah, it just was so off the wall to me. I didn't even know what to say.
I've never seen that.
Yes, thank you.
Want to connect with the community? Watch the replay and meet people on MLVX.ai